This is a uh, figure that shows some of the morphologies of cyanobacteria. And cyanobacteria are really interesting as far as bacteria because they really have a lot more advanced morphologies and a range of different shapes and functions of cells that many bacteria don't. And they have things like circadian rhythms and some special adaptations that make it so they can really dominate in situations, in some extreme situations. And as mentioning before, they were probably some of the very first organisms that inhabited a large portion of the Earth in terms of having a global biogeochemical effect. The earliest fossils look like structures that were created by cyanobacteria today that are extant, and they're called stromatolites. So earliest fossils are about 3.5 billion years ago, and the Earth is 4.5 billion years old. And by 2.5 billion years, they had oxygenated the Earth's atmosphere. And some of the terrestrial versions are probably were the first organisms that started building soil on land as well. In this figure, we can see some of those unique adaptations. And the first of these is these clear cells in this chain of cells. And these are called heterocytes, or until recently, they were changed to heterocytes from heterocysts. And so I believe it's heterocyst in the book, but you might hear heterocyte as well, as that's the up and coming term. They're the site of nitrogen fixation and allow these cyanobacteria to use nitrogen gas as a nitrogen source. So if nitrogen's limiting, that gives them a competitive advantage. And we'll discuss this again in more detail when we hit the nitrogen cycle in a few lectures. In addition to this, they have vegetative cells, and these are where the photosynthesis occurs. And they have akinets, or resting cells. So these large cells here are resistant, and if the conditions get bad, they can go settle down on the bottom of the lake and sit there until things get good, and then create a new filament, start dividing and creating new filaments. In addition to that, they have gas vacuoles, which are these proteinaceous vesicles that have a hydrophobic inner portion, so it keeps the liquid contents of the cell out of it, and they make them less dense than water, so they'll float, and you get floating cyanobacterial scums or blooms that are related to that. So those are a number of the adaptations. Additional adaptations you see here, this one right here is microcystis under H, and it has this large gel-like matrix that it's embedded in. And this makes it less likely to be grazed because the Daphnia and the zooplankton can't process this large gel blob. They're really important because microcystis is one of the really common species genera that creates toxins. It's microcystin is named after it. And they occur in fresh waters. They can be found in marine waters. They can be found in hypersaline waters. They can be found in oligotrophic water as well as eutrophic water. In a general sense, the streams, the species such as this rivularia is found in flowing water. It tends to be in oligotrophic situations, whereas these planktonic versions tend to be found in eutrophic lakes. The last adaptation that they have that gives them the partial ability to form blooms is the special pigments, the phycobilins that we've already discussed in the chapter on light. And those are pigments that allow them to absorb green light. Chlorophyll, remember, absorbs blue and red light, and then they can fill in that central part. So we saw, particularly when we pulled up the samples from the lake, from Potawatomi State Lake number 2, and there's that deep layer of cyanobacteria. That's why they can hang in down there, because they've got an oscillatoria. This is the one that it looks like. They've got these cells that can use green light, and then they can regulate their buoyancy to get right at that point where they're getting enough light, but they're also low enough that they can get the nutrients that are diffusing in through molecular diffusion across the metalimnion from the lower part And we'll see when we look at the chemistry that the nutrients are way higher in the meta or hypolimnion than they are in the epilimnion, because there's a lot of competition with the algae above. Oh, I know I need to give you the right view for this. Sorry about that. That might be better. And I mentioned cyanobacterial toxins, and that's one of the really big water quality issues that faces the world right now. That places all across the world are experiencing more cyanobacterial blooms. And cyanobacteria can produce hepatotoxins, which are liver toxins, and neurotoxins. The neurotoxins are really super toxic, and they call them, it can be called very rapid death factor, because if you inject a mouse or something with this stuff, it just takes a few moments for them to go. The hepatotoxins damage the liver, and there's more and more information that there's low chronic exposures may cause liver cancer. So in areas where there's a lot of cyanobacteria in the drinking water, the levels of liver cancer increase substantially. The first studies that showed that were from Australia, that correlation were from Australia. The problem with that particular study was that there was an area that had very large rates of alcohol consumption, which can cause the same issue. And so further studies that have factored that out have still strengthened that correlation. It's possible for the toxins to be bioconcentrated by some organisms, and we'll talk about when we get to toxicology, we'll define that more completely. But basically, if something can eat them, it can concentrate it over the background concentrations. It's thought that these toxins evolved as a protection against zooplankton grazing or any other organisms. And it makes sense because the toxins are really broad. The microcystin, the hepatotoxin, works on alkaline phosphatase, and that's ATP. It works on ATP, and everything has ATP, right? So it's a toxin that can influence essentially any organism. And the neurotoxins are very broad for just animal, and affect many animals. In addition to this, other algae make toxins that we'll talk about. A dinoflagellate, paramnesium, has been responsible for massive fish kill in Colorado. And there's been some minor, there have been some other episodes of that in Kansas reservoirs as well. And it can also be important in fish culture ponds. And then there are other toxins and harmful algal blooms in marine systems as well that have been of concern. So it's not just the cyanobacteria, but the real one that's important is of worry is the cyanobacterial toxins, because of its broad spread increase. And as people increase the nutrients in the water, the tendency for the blooms becomes greater and greater. And any summer in Kansas, if you Google by the end of the summer harmful algal blooms, Kansas Department of Health and Environment, here's the one that was updated. It looks like about 10 days ago, maybe, or 6 days ago, excuse me. And we see that we only have one warning active, but we have the levels are coming up in over a dozen reservoirs and lakes, and those include an actual river, which is a slow-flowing river that can have high levels. Milford has the reservoir near us has quite a few, and maybe some of you live near or grew up near one of these other areas. But this is really common to see. This page is pretty good. There's some a few errors if you go through the menus. There's some things that are simplifications, I would say, not so much errors, but we'll talk about eutrophication control and some of the things that people do that aren't necessarily very productive in the eutrophication lectures. Okay, so first question of the day. Why have global climate change and land use changes led to increased prevalence of cyanobacterial blooms? So we've got some good suggestions here. A lot of people got the increased fertilizer runoff right. As we feed more people globally, we have to have more crops. And this means that we're going to have more runoff of nutrients. So nutrient enrichment has been a um, common effect. And there's a pretty good evidence that above about 30 micrograms per liter total phosphorus in the water, that the probability of cyanobacterial blooms comes up considerably. In addition to that, cyanobacteria tend to do well at warmer temperatures. I already mentioned that they're adapted to extremes, and as things get warmer, then you're going to have more of them. So I mentioned just a moment ago that if you go at the end of summer and look for the warnings, that's when you see them. Well, that's because, in part, because there's the temperatures are warmer. In addition, they're also resistant to grazing, as we talked about with the toxins. And so as the zooplankton populations come up in the summer, they're the ones that can hang in there. The other one that I don't think anybody really got, but I don't expect you to at this point, is so when you get this dense bloom that's photosynthesizing, they have a high demand for carbon dioxide. And if there's more CO2 in the atmosphere, you can have greater diffusion rates of carbon dioxide into that surface layer that they're concentrated in. And that can lead to increased photosynthesis and allow them to continue to dominate the other organisms. The bloom can maintain itself because it comes up and it covers the surface, right? So what happens to the algae that are below it? What limits them that don't float up to the surface?
They don't have any sunlight.
Yeah, so they get outcompeted for the light. They get all the green light, the red light, and the blue light, right? So they can't get light there. And then the other trick they have with their gas vacuoles is that if they become nutrient limited, they collapse their gas vacuoles and they sink. So they can sink down where the nutrients are deeper down and then re-synthesize those gas vacuoles and pop back up, right? So this may happen over days or so or more, but they can move up and down through the water column by regulating their buoyancy and outcompete the other organisms. So if they can deal with high temperatures, they can outcompete everyone for light, they're resistant to grazing, and they can also outcompete for nutrients because they can go where they are and come back. This gives them a way to make really intense blooms. So that gets us through with the bacteria, and we're off into the Protoctista, which I mentioned is a sort of an artificial group. It's a huge and varied group that's sort of scattered around what we call that group. And the taxonomy that we talked about in the trees actually make that fairly clear. Still, we'll talk about the groups that are together here. So the remainder, really important primary producers in freshwaters we'll talk about, and it's really dominated by the algae in freshwaters, although macrophytes can be important as well, which we'll talk about near the end of this chapter. And these include the chrysophytes, which are flagellated and ingest particles, Bacillariophyceae, the diatoms, which have a silicon frustule, and it's useful in paleolimnology and forensics, and it can be found in many types of freshwaters. The dinoflagellates, Dinophyceae, and some are toxic and cause fish kills. The euglenophytes, Euglena, and the chlorophyceae and charophyceae. So the green algae and then the charophytes, which are the progenitors to land plants. So we can talk about some of the groups here. We have chrysophytes. Dinobrion is probably the most dominant chrysophyte that we'll see in lakes around here. And this is one that we see late on because it's really quite large. And it's an interesting sort of candelabra shape, and each of these individual flagellated cells somehow coordinates with all the other ones that are stuck together in this thing. And this thing's about as big as a small zooplankton. So when grazing pressure goes up, these things can start doing well. And they can go up and down in the water column also just by swimming. And then we have Batrachospermum. So the red algae are much more common in marine waters than fresh waters, and they also have those same pigments that the cyanobacteria do that use green light, and they can be adapted to very low light, so they can be found quite deep in marine environments. But Batrachospermum is a pretty common one in streams in the world, not so much around here, but more cleaner streams. I have a, do they produce toxins like cyanobacteria? Which group were you talking about, Lane?
The Batrachospermum.
Probably not. Although my guess is that most species have some level of toxin that they're producing. It seems pretty common, just like most plants have secondary compounds that can influence the things that graze on them. But it's not as widespread of a problem as it is for the cyanobacteria and some of the dinoflagellate species. The next group to talk about is the diatoms. And this is a really beautiful group. Here's a bunch of different morphologies of the diatoms, and we'll see these in both the lake samples and the stream samples that we have collected. And diatoms, the sort of key diagnostic feature is the fact that they have this glass cell wall. And it's best illustrated by the one on the bottom here. This is one of what's called a centric diatom. So if you look at it from the top, it's circular. But if you look at it from the side, you can see it's like a petri dish. And the pieces come together like that, right? And actually, it turns out if you look at any of these from the side, this A through E, they'll also look rectangular. So they'll almost look like this. You know, if you just turned it up a little bit and couldn't see that it was a circle behind there, they'll look rectangular because they have the two pieces that fit together. The characteristics that are used for taxonomy are the size and shape of the entire cell wall, the frustules they're called, and also the striae, which are these lines, how many striae there are per 10 microns, and the puncti, the holes that make up the striae. In some of these images, you can see the holes really pretty clearly. So you have a silicon frustule around your entire cell. What's the importance of having holes in that?
Respiration.
Yeah, diffusion, respiration, everything. You know, it's like if you put us in a glass jar with no holes, you know what happens eventually, right? We're gone. So they have to exchange with the outside world, and that's what they do. In addition, the ones that are not circular shaped, so these are centric diatoms, these are pennate diatoms, they have this little line right here. It's called a raphe. And that actually allows them to be on solid surfaces and glide. So they can actually physically move on solid surfaces as well. Some of these species, like this Asterianella, this really common planktonic species, and it's floating, and they tend to do really well in the spring, colder water before there's a lot of grazing pressure. And they have a spring diatom bloom in many lakes, which then is replaced later on by the dinoflagellates and the cyanobacteria later in the season as the grazing pressure comes up. This one down here, K, is called Melisira, and it's actually similar to these other centric diatoms, except for this part right here on the side is expanded way out. So this is like actually a bunch of barrels that are stuck together end to end. And if you remember early on when we talked about sinking rates of different plankton, Melisira was one of the ones that we were discussing. And by adding on cells this long, they can increase their volume without increasing their sinking rate because they're increasing their surface area at the same time, right? And they're also not changing their surface area to volume ratio if you just keep adding on this way, not very much. And finally, they increase their ability to resist predation because these big long filaments, the zooplankton can't really get them. So I mentioned that these are important in paleolimnology. In paleolimnology, they'll take a core of a lake and go back in time and look at the chemistry, and they can use the remains of the diatoms to see what the conditions were in the lake over time. And they can do this because they can go out and look at all the lakes now and say, okay, what's the pH in all these lakes? What diatoms dominate? And they can figure out what community is characteristic of a lake that has a high pH or a low pH, or is a high temperature or low temperature, right? And they can go back and look at that through time, and you can core, and then you can get a isotopic concentration. And going back to a student mentioned carbon dating, you could do carbon dating up to about 20,000 years, or use some other isotopes and get that. And this has been a really important tool in looking at things like acid precipitation, where people said, oh, well, these lakes were always acidic. Well, no, they weren't necessarily. So why do you think these diatom frustules are particularly useful in this particular application? Why do you think they're well preserved in the sediments?
Is it because they have a tough outer structure?
Sorry, Trevor, I can't quite hear you.
Is it because they have a tough outer structure?
If you're still quiet, maybe you can click to type it in. I'm sorry, it just, your microphone wasn't coming through. I think Cassie got it though. They're glass, right? So glass doesn't break down very fast. It really dissolves slowly, so they can just be preserved for a long period of time. The other interesting thing that they're used for is in forensics. And so when a person drowns, one of the last things they do is take a breath of water. You know, you hold your breath for as long as you can, and then you have to breathe. Your body, once you pass out, your body will breathe water in just automatically. And as it does so, the water goes into your lungs, and it breaks your alveoli, and the lake water actually gets into your bloodstream. Then it goes through, as your heart beats before it stops, it pumps the diatoms through your entire circulatory system for a moment or two, a few minutes. This then means that if they want to find out if somebody had drowned, what they'll typically do is they'll take bone marrow from inside of a bone, because the circulation can get the diatoms in there, take that out, put that in acid, and eat away everything else and see if there are diatoms in there. And they can then use the diatoms to link where the drowning happened, or if somebody drowned. So one of the things that could happen is somebody could have got bumped over the head and killed, and then they'd be thrown in the water. And it'd be important to know whether they were killed outside of the water or if they just drowned. And if they don't have diatoms in them, then they wouldn't have drowned. Something else would have got them. There have also been cases where somebody's been drowned in one spot and then pulled out and thrown somewhere else, like, you know, drowned in the backyard pond, and they pulled them out and threw them in the river. Well, they could find that the diatoms that were in their body were related to where they were drowned and not in the river. In this particular case happened in England, where people have been working on phycology or algology for a long time, and they knew exactly which pond had that kind of diatoms in, so they could trace it back to that. The other thing that could happen is if you find a body part, you don't know if, you know, the person was drowned in the water or whatever, but if you had found an arm, you could still get the bone marrow out of that arm and boil it down, and the diatoms wouldn't have gotten in there. They would have to circulate with the person was alive and find out, well, you know, maybe the person drowned and then an alligator ripped their arm off or something like that, right? So you could use it that way. And then they've also been used. There've been some cases where there was a case where a young man was beaten by a group of other men, and they claimed that they had never been to the site where the beating happened, but their shoes were sampled, and the mud that was on their shoes had diatom frustules in it. They were able to link it back to the particular site that it happened. So that's some interesting, an interesting fact that people tend to remember. And we'll move on from the gruesome to some other groups. And the top group of this is Euglena. Euglena is a really interesting organism, and this is, sort of where microscopy and even molecular taxonomy blurs the line between the old line between what were animals and what were plants, right? Because these are photosynthetic organisms, yet they're modal. In addition to that, they're phagocytic. That is, they can engulf things, right? So they can eat like bacteria, like an amoeba would, and they have amoeboid motion as well. And then to add on to this, they're mesokaryotic in that they have a nucleus because they're eukaryotes, but they also have extra nuclear DNA that's out there, so other chromosomes that are not inside the nucleus. They think this is related to ingestion of other organisms that became endosymbiotic. The euglenophytes are really commonly found in eutrophic waters. So they don't necessarily need to photosynthesize, and then they can swim to areas that have light when they need it. So they can be positively phototactic. And you can see there's this little dark thing that's actually a red spot. They actually have light sensitivity, so they can move in and out of the light as they with phototaxis. Again, we'll talk about that when we talk about motility of organisms. We will see both these morphologies of dinoflagellates down here, the next group we're talking about. And these are interesting because they have a flagella that goes around that's inside of this groove, either this groove or this groove right here. And then there's going to be another groove, which is behind here and is down here, where another flagella comes out. So they are actually a modal organism. As I mentioned, the toxic dinoflagellates, the one that looks like this it can be toxic. And in marine systems, the Chesapeake Bay, there's been a real problem with toxic dinoflagellate blooms because they make a neurotoxin, and the fishermen that worked there had permanent neurological damage, as well as some of the people that were initially working on the organism in the lab before they understood that just the toxins coming up through the air could give them neurodamage. These organisms are quite interesting because they also straddle the line between photosynthetic and non-photosynthetic organisms. Some of the species don't have any chloroplasts and live completely on engulfing other materials, but yet others do. So when we talk about the protists, there's this strange mixture where you have some of these colorless organisms. They because of this ability to move around and be toxic, and then also because these are fairly large in morphology, they can be resistant to grazing, and so we'll see in the lakes that we have sampled this summer that this is a really, really dominant type of alga that's in there. The bottom part is charophytes, or the stoneworts. And this is thought to be the progenitor to land plants because we start getting algae and they actually are encasing their gametangia and sterile cells, which then allowed organisms to move out of the water onto the land and keep their gametangia from drying out. And they're more complex than most of the other algae, but they're seem somewhat related to the green algae, although less so once the molecular techniques have come out than was thought previously. Jaden said, if you accidentally breathe water when you're coughing, is that enough to have diatoms in you? Yes, I think so. Yeah, you'd have a few diatoms floating around in you. So that's definitely true. But, not a lot. Not like when you drown, where you really get a lot. Okay. Next, we're talking about the green algae, and the green algae are really dominant producers in freshwaters. There's such a huge variety of types. You can have single cells, like this Chlamydomonas over here, or if you put a bunch of single cells into a giant ball, you've got volvox. And this is what people are using to get at the steps towards multicellularity, whereas this is kind of an intermediate where these things are sort of coordinated, but it turns out they'll go from this form to this form in response to predation. You can have large filamentous forms. This Cladophora is the probably one of the most dominant stream algae that you'll see, and it's really in freshwaters everywhere. Spirogyra has the spiral chloroplasts, very common. Lane?
Are they multicellular, or is it like several different organisms coming together?
They're multicellular in all these cases. So this particular one is a single cell. They can be multicellular or versus single cells. These Volvox are basically a multicellular from dividing single cells. They haven't come together from outside. That's a good question. There's an interesting one here. This one, Basicladia, is found mainly on the shells of turtles. So if you see a turtle with green algae streaming off of it, it's almost certainly Basicladia, and that's almost the only place you'll ever find it. I think people have found it a few other places, but it's a weird and interesting adaptation. So the amount of morphological variation in the green algae is really impressive. I will also say that I've got an independent mini lecture on algae for identification that's associated with the lab that we'll be doing next week, I believe. So this is just a single group of green algae, the desmids, and this shows you the kind of morphological variation that you can have in one group. We can go from this star-shaped thing to a relatively simple thing, and this would actually be on two sides. I think there's two groups of two species of Cynodesmus that we'll see in the samples that we have here. Very common in eutrophic waters as well. Again, these spikes are probably to keep from grazing, but they'll also lower sinking rates. And this Pediastrum is one that is one of my favorites just because it's such a beautiful one. And we have one that's sort of like this in the samples. It just has an open area here. But they can make chains. So each one of these is like one of these cells. So they can be multicellular in terms of being filaments or single cellular. So we'll move to the protozoa. Protozoa are really important consumers of bacteria, and they can drive the microbial loop. Some can even eat viruses out, the particles as small as viruses. So it can be really important in the cycling that's happening with the bacteria and keeping things going around. The Mastigophora is the flagellates. So some of them are flagellates and some of them are amoeboid. The Phytomastigophora are green and the Zoomastigophora are colorless. The thing that I talked about, like with the dinoflagellates, is that you can have things that are fairly closely related and have dropped their chloroplasts or obtained your chloroplast over time. So this is kind of a weird separation. It's not always necessarily an evolutionarily correct one. The sarcodines are the amoeboid protozoa, and that's the blob that goes out and eats everything. It's not going to be swimming around freely as easily, but it can crawl well on sediments and such. And the Coeliophora, the ciliates, which we've seen as paramecium being a really common lab animal that maybe you saw in intro biology or organisme. So the ciliates, Coeliophora is here. Here's a one that looks like Euglena, but it has that single flagella, an amoeboid protozoan, and some nice ones, Vorticella, which is attached on. And these are the ones that you'll see that come down and then pop up and filter and come down and pop up and filter. So a lot of these are heterotrophic organisms. The fungi are important as well, and there are aquatic and terrestrial fungi. The aquatic fungi can be, they're saprophytic for the most part. That means they're breaking down organic material, dead organic material. And they're really important in degrading leaves and things that fall into fresh waters, really important in streams. Some of them are predatory, and we'll talk about those in a little bit. The lichens are also found in fresh waters. There's symbiosis between a fungi and an alga. And the fungi basically creates a structure on the outside, and the alga so protects the alga, maybe brings nutrients into it. The alga photosynthesizes and provides organic carbon for the fungi. They can be really, so it's not only a symbiosis, but it's a mutualism. They can be quite important in some wetlands. The plants then can be divided into vascular and non-vascular plants. And in a general term, a catch-all term for these is macrophytes, that is plants that you can see with your eyes. But large algae can also be considered macrophytes as well. So things like Chara or Cladophora, the large filamentous alga, can sometimes be considered a macrophyte. So macrophyte's not really a taxonomic term, but just means large plantish thing if you look at the roots. The roots of the word, the origins of the word, not the roots of the plant. They have different growth habitats, and we already covered this a little bit when we were in the wetland section, but they can be emergent, which means that parts of the plant stick out into the atmosphere. So this would be things like bulrushes and cattails, for example. They can be floating attached. So the water lilies oftentimes have roots down in the bottom, and they can get big enough that the Amazonian ones that you can lie on one. So they can get quite large. They can be submersed, completely submersed, or they can be floating and unattached. And so duckweed, which you've seen, I'm sure, in ponds around, and another one, important one, is azolla, which is an aquatic fern that has a cyanobacterial endosymbiont that we'll talk about when we get to the mutualism chapter, or also important in some systems. So if we go to Tuttle Creek Reservoir, we didn't see any macrophytes in that reservoir. And we, in pot 2, had a lot of macrophytes in the shallow areas. So why do you think it is, which is a much smaller lake reservoir, and it also tends to be more stable in that it's not, the water level of that reservoir is not very regulated. It just, if it more flows in, more flows out, whereas Tuttle, it is used for flood control. So why do you think it's more difficult for macrophytes to establish in large reservoirs? And I think what I'll do